Welcome to Library Seminars, NOAA Central Library's webinar series for the presentation of research and ideas in support of the National Oceanic and Atmospheric Administration's mission. Today's seminar is the first for the 2022 Knauss Fellows cohort. The Lunch and Learn series is a monthly webinar where Knauss Fellows showcase their own research or the projects they're supporting during their fellowship. Teresa Vander Woody, Knauss Fellow at the United States Environmental Protection Agency's Nutrient and Biocriteria Program, which is part of the Office of Water, will be our host. She will introduce our speaker, Alfonso, Macías, Tapia. But before I turn this webinar over to our speakers, here are just a few tips to help you enjoy your live presentation. If you're having trouble with the audio or the visual components of GoToWebinar, please log out and rejoin us. This will reset the software and resolve most technical issues. This presentation is being recorded and will be available on the NOAA Central Library YouTube channel later today. And I'm going to put the link to the channel in the chat box as soon as I finish talking. We highly encourage you to ask questions, which our speaker will address at the end of his presentation. So type your questions throughout the seminar as the questions come to you. But the question chat box is located in the GoToWebinar control panel, which is most likely on the right side of your screen. So I'm going to now turn the presentation over to our host, Teresa. The mic is all yours, Teresa.
Thanks a lot. So I'm going to introduce Alfonso today. Alfonso is an oceanography PhD student at Old Dominion University, where his research focuses on the water quality effects of extreme tidal flooding events. Since getting his undergrad and master's degrees in Mexico, he's worked on topics from wastewater treatment to natural water systems, how they work, and how to preserve them. He's also part of the Virginia Science Community Interface, which is a coalition of scientists and engineers dedicated to getting science into the hands of community members. He's a 2022 Knauss Fellow in the NOAA Office of Education, working on science education and policy. We're looking forward to hearing more, so I'll turn the floor over to Alfonso.
Thanks, Teresa. Welcome, everybody, and thanks for attending the talk. Also, thanks for the Lunch and Learn seminar organizers for the space to present. As you heard, my name is Alfonso Macías Tapia, and I'm a 2022 Knauss Fellow for the NOAA Office of Education. You also heard that I also wear two other hats, very proud of. I'm a PhD candidate of the Ocean and Earth Department at Old Dominion University in Norfolk, Virginia. And I am also the recruitment chair for Virginia Science Community Interface, this grad student organization that brings science to advocates. So I'm talking to you today using my grad student hat. The title of this talk, this is like the more technical title that I use for scientific presentations, is 5 years measuring the Muck, evaluation of extensive tidal flooding, biochemical characterization. But in other words, it's just seeing what is happening in terms of water quality when tidal flooding events occur. So the content is part of my thesis dissertation and also part of the paper that we have published and those that we're currently working on. So before I dig into the content, I would like to thank the Hampton Roads Sanitation District for the funding to do this project, and also thank the collaborators that make this work possible and more rich. Sophie Clayton also from Old Dominion University, from the Oceanography Department, Corday Selden, who's a postdoc at Rutgers University, Tom Allen, also at ODU, but in the Geography Department, Derek Loftis, who is at Virginia Institute of Marine Science, Pete Bernhard, who is the lab manager for our lab, and Margie Mulholland, amazing PhD advisor. So this is the overview of the talk. First, I will give you an introduction about sea level rise and its connection with water quality. Then I will describe the citizen science approach that we're using to characterize this non-point source. Moving into the results, I'm going to describe to you the variation that we have found in the 5 years of the project. Then I will describe a comparison between floodwater concentrations and nutrients in floodwater concentrations of nutrients in floodwater, versus a standard, which I'm calling like a background, different forms of points of reference to see whether or not those concentrations are high or low. Spoiler alert, they're higher than those points of reference, so we're going to evaluate this potential of tidal flooding events as input to the ecosystem. And at the end, if I have time, I'm going to focusing on a model that I'm trying to build based on correlations. Like, are those concentrations higher where there is like grass, for example, or where there is a pavement, where there is cars, etc. So we're going to get to that later. So let's just start with the connection between sea level rise and tidal flooding. So sea level rise has resulted in increases in average water levels during high tides in many coastal areas around the world. So let's explore that using this figure from NOAA, which shows a hypothetical coastal city. You have an apartment building, let's say, and then a store next to it, next to the water. So in 1950, you have in the darker blue the water level, the average water level in 1950. So in 1950, it will take a considerable amount of water caused by a large storm, such as a hurricane, to cause high tide flooding. So this was like the average water level. Probably high tide will be somewhere here, and then if you get a storm surge, those were the times when you would probably have some impact, minor impact, but nonetheless impact on the coast of this hypothetical coastal city. But then we have sea level rise. So in 2010, with higher relative sea level, it no longer takes a stronger storm or a hurricane to cause flooding. Now, high tide flooding is frequent and can cause merely just by high tide. But now this is the average, or not now, but in 2010, this is the average high tide, the average water level, and then with high tide, you will have these minor affectations. And then if you put the storm surge that they were using in the other example, if you put that on top of the new sea level, of course, the affectations just become more dramatic. So this is happening in many coastal cities around the world. One of the affected areas is our study site, the city of Norfolk, Virginia, located in the northeast of the US. This region is in the Atlantic side of the continent, in the lower part of the Chesapeake Bay. You can see it on the map. We're located in the bottom of the Chesapeake Bay. We are part of the Lafayette River crosses through the city, and then it connects to the Elizabeth River, which connects to the James River, which connects to the lower part of the Chesapeake Bay. So here's our university, and that becomes relevant because this is the point where samples are going to be brought during our sampling events. So as shown in the map in black, when tidal flooding occurs, the water from the Lafayette River inundates the urban areas. As mentioned before, this problem is more relevant because as in many other parts of the world, the intensity and the frequency of these events is increasing. For example, the graphic on the top is showing maximum daily water levels near the mouth of the study site. There is a station near the mouth, somewhere up here, and we have data for almost 100 years. It goes from 1930 to 2020. So colors show different scenarios, from the green color indicating non-flooding conditions to the yellowish color in the middle shows like minor flooding events, like the one in the hypothetical city or in the hypothetical coastal area in which water is touching the streets and the stores, schools, etc., but it's not as dramatic, still causing affectations. And the red dots indicate extreme flooding events, and you can see how the most extreme types of events get repeated more and more often by the decade, and how the intensity also have a positive trend. So you can see how these red dots, just to retake it, are becoming more frequent and more intense. Like every time you can find dots higher in the graphic and repeated like in a more constant manner. And if you're not familiar with the topics, you're lucky not to live in one of the cities that are affected by this. Here are some examples of pictures collected by our volunteers during the different sampling events, just to give this data a little bit more of a human side of like the affectations that this causes. So the houses, some of the streets, the way they look like, the roads and the cars. No idea how many cars we have seen stuck in the flooding. This gigantic truck even has issues passing through. So yeah, now I hope this figure will look a little bit different. It's just not data and graphics and how this sea level rise and flooding events are increasing, but the affectation that they can cause to the people living there. So due to the impact that tidal flooding causes in coastal cities, as you saw in the pictures, the physical and economic effects are well documented. However, little is known about the chemical and biological effects that this event, the tidal flooding event, can cause in adjacent aquatic environments. So just to give you a better idea of the reasoning behind the project, I'm using this my incredible artistic skills to show you what is happening. So terrestrial system accumulate various type of organic and inorganic matter. I don't know, we have the plants, the stuff coming from the cars, stuff coming from the house, the trash, pets, like all the type of things that you can imagine that lay on the perimeter of a coastal city. So then when the floodwater comes and inundates all that area, it's going to get in contact, and either through diffusion or advection, it's going to carry some of that stuff. When the floodwater, like this is the high tide in number 2, but then in number 3, then we have low tide, so the water retreats back to the estuary, and when that happens, we think that it's bringing some of that back to the ecosystem. So we want to figure out what is it that it's bringing and how much it is that it's bringing. So if you don't believe my amazing artistic skills, here are some like actual pictures. One of the pictures. Again, we have many from the volunteers and from the different samplings that we have done. Diapers, all type of trash, toilets, dead animals, we have everything. But for respect to the audience, I'm showing one of the ones that are like nicest, but it still shows the point. The trash and many things are laying on the ground. Flood water comes, gets in contact with it, and then it retreats back to the estuary. So how we have addressed the water quality characterization of tidal flooding events? Well, while trying to do this, we realized that we needed more people collecting samples to cover a representative area in the short amount of time that the floodwater inundates the street. Depending on how intense the flooding it is, it can only be a few minutes to 20, 30, probably to an hour flooding. So in that time, we need to collect many samples. So of course, we need more people. So to solve this, we invite people from the public interested in collecting water quality samples from tidal flooding to an event that we name Measure the Muck. That's why the title of the presentation of 5 years measuring the Muck. Volunteers range from kids in high school or even younger, those from elementary school, to their parents, their teachers, as well as professors and grad students, mainly from our university, from Old Dominion University. So before the event, they come to the lab and they get training how to collect water samples. We also use this opportunity as a way to show them around in the labs, show them the instruments, talk about water quality, ecosystem, oceanography, all those type of things. Normally these young audiences and also the parents and the teacher get really excited. So we use that opportunity to also show them around. The event, one important characteristic is that the event is done once a year around the fall, perigean, springtime. This is when the moon and the sun are aligned to the earth and to the closest point to it. So they are, what's the word in English? Making the strongest gravitational force. The nature of this phenomena makes it very predictable and allows us also to plan ahead, many months ahead, when we're going to have this event, and we can coordinate with the volunteers to do it. So the day of the event, the volunteers, they have been trained and everything. So the volunteers come to the lab and they receive a sampling kit that includes a cooler, an ice pack, sample bottles, gloves to protect the volunteer and also the sample, a board to write notes, coordinates, time of collection, data about like the land use that it was around the sample, and also a mapping indicating where they're going to go. We try to make teams of different ages and different education level, and then we send them to, so they gain a little bit of different knowledge, not just the experience of the sampling, but they can also talk with other people, and then we send them to different areas that we have previously planned. Another important element during the sampling is the use of a phone app. It's called Sea Level Rise, and with this app, among other things, our volunteers can take pictures of the site to show us if there was an outstanding amount of trash or something very relevant for them to show us. Another thing really important is that drops a pin, a GPS pin, that is going to tell us where exactly the volunteers collect the samples, and then we can use that to process the data. When the samples are collected, they're brought back to the lab and we process them for later analysis. In previous years, multiple volunteers stayed to help us process the samples, and the lab looked like the images on this slide. Of course, because of COVID, last couple of years has been a little bit different, but we have still been able to manage to have some of the volunteers, especially those that have repeated for multiple years. So yeah, if anyone is interested, I'm not going to focus more on this citizen science piece, but if anyone is interested, well, I can talk more during the Q&A session. So far, sampling events have occurred in each fall from 2017 to 2021. Last November, we have our 5th event, measure the Muck 2021. And each year, we have had the minimum amount of volunteers has been somewhere around 30, and the maximum has been somewhere around 60. Of course, there's fluctuations in the years. The past couple of years, because of COVID, also become harder to plan. But even with those fluctuations in the volunteers, we have been able to collect about 200 samples each of those years. So this is the map showing the distribution of the samples in the Lafayette River and the perimeter of affected areas of Norfolk. So multiple parameters have been measured for these samples, from dissolved nutrients, which are the ones that I'm going to focus during the rest of the talk. But we have also collected samples, for example, about fecal indicators and polycyclic aromatic hydrocarbon. So if you have questions and interest about these two results, fecal indicators, for example, is a really interesting results that we have. But for the sake of time, I'm only going to focus for the talk in the part that my dissertation and my research interest is focused on, which is dissolved nutrients. But again, if you have questions about the others, please ask later. Okay, so we got ourselves an army of citizen scientists and swipe the perimeter of the affected urban area. So how do we know if that's enough? Like, do we need a bigger army? Do we need to collect not 200 samples, but 1,000 or 5,000 or 10,000? How many samples should we be collecting? So to measure the variability of the system, we will use the behavior of the mean and the standard deviation. And to explain this idea, I will use data for dissolved inorganic nitrogen collected during the 2021 event. Then I will show you the results of the rest of the year and the other parameters like phosphate and dissolved organic nitrogen. So pretty much what you do is you take like your 200 samples and put it in an imaginary black box or black bag. So you mix those 200 samples, you take 5 samples and collect random samples that you mix. You don't know which ones they are, so you take 5. You calculate the mean and the standard deviation. Put them back, mix them again, and then take 10. Put them back and repeat the process until you complete your maximum amount of samples. For our case, is 200. So then you put those 200 samples back again, you mix them, and then you repeat the whole process again and again and again and again. With the magic of coding now, this is a very simple procedure, and the principle tells us at the end, the message that I want to give here is like the principle tells us that if we have had collected 500 samples and 1,000 samples, you can see how the mean and the standard deviation from these like random collections it gets reduced the closer we get to the 200 samples. So what this indicates is like if we will be able to collect 500 or 1,000 or 10,000 samples, we will only reduce that mean and that standard deviation. So in some sense, this indicates that for like 170 to 200 samples, it seems that we are capturing the variation of the systems in terms of dissolved inorganic nitrogen. So I'm not going to stop for a long time in this type of slides where I present you the rest of the years. Because I just want to give you the general message. Like if you have specific questions about the different years, this is something that catches your eye, you can ask later. But here I just want you to take the general message, which is like it seems that for all of the years that we collected, in this case dissolved inorganic nitrogen, that variation gets reduced the closer we get to 200 samples. So that seems like a very good number, which is really relevant not only for tidal flooding research, but other studies of non-point sources. It's having an idea of how many samples are enough. Like the comment kind of joke that I was making at the beginning, that is like, is our army enough? Do we need like a bigger army of citizen scientists to collect 10,000 samples? So again, I have the results for the other years, but in general, they look the same. So now that we saw that we capture the variability of dissolved nutrient concentrations in floodwater, let's move on and use that data for comparisons. Here we will compare between floodwater, the results from floodwater, and what I call background, which is just like a point of reference from non-flooding conditions in the estuary, different forms of a point of reference from the estuary. So I will explain to you those different points of reference. So first, in red, I'm showing you the histogram of dissolved inorganic nitrogen in micromoles per liter for the 200 samples collected in the 2021 event. And I'm going to keep using the dissolved inorganic nitrogen data in 2021 for the other results that I have, just for the sake of explaining the concept. And then again, I will give you the take-home message, because for the other years and for the other parameters like phosphate and those dissolved organic nitrogen, results look very similar. So I don't want to get you twisted presenting like the 100 figures for all the years for all the parameters. So let's just focus in one. So there is dissolved organic nitrogen for 2021. This is for floodwater. The red one is for floodwater. And then you add the data for the long-term data. In this case, this data comes from samples collected between 1998 and 2018. And probably for people who manage other type of data, 1998 to 2018 is not like a long-term series, but in biochemical water research, I will consider that a long-term data set. So we have that data set, and then we only use the data from the months of October and November, because as I mentioned before, we're focusing our sampling during the fall perigean spring tide that in the region in Norfolk, Virginia occurs at the end of October or in November. So in blue now you can see the results from the short-term sampling, and these are just samples that we go and collect on the mouth, from the mouth and the head of the system. This is a project from our lab in which we're trying to see the triggers of harpoon alga bloom. So we are continuously collecting nutrients and other forms of data, temperature, oxygen, chlorophyll, etc., to see how these blooms develop. When do they start? Why do they start? The conditions under which they are starting and how they thrive. But anyway, from this project that runs mainly late spring to early fall, I'm taking some of this data, the data that is closest to our event, and use it as another point of reference. This one is like more continuous. The long-term one only has monthly data. This one has like sometimes even daily data. The most recent years has become, the sampling has become even more frequent. So last form of background, the last form of point of reference is a boat transit. We literally, as the name indicates, just take a boat and go from the mouth to the head of the system and collect samples to see what is like a normal nutrient concentration throughout that transit. So you can see, that comparing the floodwater overall, these floodwater concentrations, at least visually, and we have a statistical results for all of that, but at least now, just believe that piece is higher than the rest, the three different backgrounds, the three different points of reference. And results look similar for the other years. This is still dissolved in organic nitrogen, but for the 5 years that we have done the experiment, I have also results for phosphate and for dissolved organic nitrogen. But again, for the sake of time, just stick with the general message, which is in general, floodwaters tends to be higher than the different backgrounds we're using for comparison. So the above, as I mentioned, is a graphical representation of the difference. We have like a full statistical analysis if you really want to know the details of that piece, as during the Q&A session. So based on those differences between the floodwater and background concentrations, now we're going to discuss how much of an input, now that we see that floodwaters tends to be higher than the point of reference for the different years, for the different parameters. So how much of an input do they, now we want to see how much of an input they can represent to the ecosystem, nutrient input they can represent to the ecosystem. So to calculate this input, we will use 1, the differences between floodwater and background concentrations, and 2, the volume of floodwater during a tidal flooding event. So let's start with the difference in nutrient concentration. Where floodwater concentrations were statistically higher than background, we use those values to calculate the difference. Those are the only differences that we calculated. Those were statistical significance was found. So once we pass that statistical significance threshold, we move into the difference. We create two different scenarios. Like one is like a high scenario, a dramatic scenario of differences, of nutrient differences. In this case, I'm using the mean of the flooding plus its standard deviation. So if you see the graphic on the middle, it would be like the mean of that histogram plus the standard deviation of that calculation. And then we're subtracting the value, the mean of the background, in this case, the long-term data, minus its standard deviation. So that difference is like the biggest difference between these two data sets that is possible, let's say. We repeat then the process with the rest of the backgrounds, with the rest of the points of reference, the bold transect, the short time series. And then we do a similar analysis, but now for like a more conservative, a low end of the spectrum scenario. In this case, we're using the mean of the floodwater results minus the background plus this standard deviation. So now this value, the background value, becomes way higher. And when you subtract it from the flooding, of course, you're going to get a smaller difference. And that's what it makes it a more conservative scenario. So this is just to show you a little bit of how this difference look, but I'm not going to focus on this. At the end, the way that we're using these values comparing to federal standards of water quality, that's when it becomes relevant. But for now, I just wanted to show you a table like how some of these values look like. And of course, the one on the right, which is the lower scenario, the more conservative scenario, has smaller numbers compared with the more dramatic scenario. We have for the different backgrounds comparisons, it would be floodwater minus boat transect, it will give you this result. Floodwater minus short series, and it will give you this result. And then floodwater minus the long series, and it will give you these results. Again, I'm focusing in DIN for the year 2021, but we have results that look very similar for the rest of the years and for the rest of the dissolved nutrients. So now we have the nutrient value. Let's get the value of tidal flooding, like a volume of flooding. For this, people who knows about this follow up, and if you have questions, again, I invite you to ask later during the Q&A. But we use depths that were extracted from the GIS raster by computing the differences with LiDAR, derived digital elevation model, and the water surface elevations for those locations where water level sensors are available in the region. There's like some along the perimeter of the system. So those differences were used to correct like the inundation depths extracted from the GIS raster. So far only data for the 2017 tidal flooding event has been processed for these calculations of volume. So for now, this is the one that we're going to use for the rest of the years. So as I mentioned before, if you have questions on the details of this, you can ask later, but you can also visit the two papers that describe in more detail this calculation. But for this talk, let's just focus on the result of those calculations that in the 2017 event is 3.9 times 10 to the 10 liters, and a standard deviation of 1.6 times 10 to the 9. So based on those calculations, based on that volume and the standard deviation, we calculated also two scenarios, a high volume scenario and a low volume scenario. And this one is very simple. For the high volume scenario, we're just taking the value minus the standard deviation plus the standard deviation. And for the low volume scenario, the less dramatic scenario, we're using the volume minus its standard deviation. So now we have the two scenarios for nutrient concentrations and the two scenarios for tidal flooding volume. So we will use this to calculate a dramatic high scenario and also a more conservative low scenario. So these are the results that we obtained for the 2021 data, again with dissolving organic nitrogen. with the comparisons with the different forms of background. The inputs in this case are already transformed to tons of nitrogen per event, per floating event. So with the highest scenario goes in the 9, 8, 12, and then in the lowest, closer to 1, 2, and 6. But again, this is not really the number that I still want you to focus on, because like how much is this much? Like 12 tons sounds like a lot per event. 0.75, like almost a ton almost a ton of nitrogen in a single floating event. It still sounds like a lot, but how much is too much? So to put this in context, we will use limits established by federal regulation. So on December 29 of 2010, the U.S. Environmental Protection Agency or EPA established the Chesapeake Bay total maximum daily loads, or TMDLs, which is a historic and comprehensive pollution diet. This TMDL includes accountability features to guide sweeping actions to restore clean water in the Chesapeake Bay and the region's streams, creeks, and rivers. So tables within this document indicate what is the limit of different forms of nitrogen inputs. They also cover, for example, solids or phosphate. But of course, for this talk, because I've been guiding you through the dissolving organic nitrogen, we're going to focus on the nitrogen table. So for example, this table 9.91, shows the values of nitrogen for our study site, the Lafayette River. It shows other systems, but I blurred them so you don't get lost in all those many names. And we're focusing now in our system, the Lafayette River. So values in this table are expressed in pounds of nitrogen allowed for an entire year. Later, we're going to transform it to tons to compare them with the values that we calculated before. So the first column shows the limit for waste load allocations, meaning point sources like those from industry. The second column shows the limit for land-based load allocations, which will mean the non-point sources, and it will include those like tidal flooding events. And the third column is for atmospheric deposition. The fourth column is the summatory of the three before, so it's just the added values of the three other columns. So now these are numbers that I really want you to pay a little bit more attention. Because based on the numbers from the TMDLs, from these regulations established by the EPA, this is the percentage that the inputs we calculated represent for the Lafayette River. So we have results for other years and other dissolved nutrients. I'm going to keep repeating this the rest of the talk, but let's keep focusing in dissolving organic nitrogen for the year 2021, just to make it clear. Again, the rest of the years and the rest of the dissolved nutrients look very similar. Sorry. So for the lowest scenario, we see that an input of 1.5 tons of dissolving organic nitrogen represent about 40% of the land allocations allowed. And compared with the total, you remember there was like a summatory of all the allocations. So compared with the total, it's a 3.7%. So when compared with the total, it doesn't sound like a lot, but as I'm going to remind you in a bit again, these are just inputs for a single flooding event. And then, of course, if we move to the higher scenario, the more dramatic calculation, we found that an input of 12.5 tons of nitrogen per flooding event represent, even already past the threshold, so represent 71.2% of the land input allowed, and then for the total, it goes all the way up to 31%. And in case you forgot, these calculations are for a single tidal flooding event, while the EPA limits are for the entire year. And also, you have to remember, as we saw at the beginning of the talk, that the frequency and intensity of these tidal flooding events, it's increasing. So therefore, failing to consider nutrient fluxes resulting from tidally induced inundations could result in underestimate of nutrient inputs into coastal water. Their exclusion could bias water quality model projections of nutrient loads to coastal areas, and this will jeopardize mitigation, conservation, and restoration efforts. So in other words, we need to put more attention into this type of issue. So with that, I don't want to make it that long. I still have the results or the piece of the talk about the model, but I'm just going to cut it here. If anyone has questions about that part, how we're finding correlations with land use, you can answer in the Q&A. But I think just to not make it as long, I'm just gonna leave it here. There is my email. So yeah, thanks for the attention, and I will be more than happy to answer any questions.
Thank you very much, Alfonso. This has been really interesting. Audience, we have lots of time for your questions, so take the opportunity to ask them live. To do that, you just have to type your questions in the questions chat box, which is in the GoToWebinar control panel on your right, and I will read them to Alfonso. While we wait for some questions, I want to remind you that we are recording today's presentation, and we'll be uploading it to NOAA Central Library's YouTube channel in the next few hours. So if you'd like to share Alfonso's work with a colleague, we encourage you to visit YouTube and share the recording's URL later on today. And also, just a quick note, I appreciated that you shared that you have published work, and the 2 articles in the Water Resources Journal, as well as the Journal of Marine Science and Engineering, can be accessed through NOAA Central Library. So if you are interested in accessing these articles, please let me know. This is Lisa Clark, and we'll be happy to help you out. So Alfonso, I just got our first question. The question asks, what is the duration of these flooding events? Are they quick pulses, or is the flooding sustained for multiple days?
Let's use this graphic. I could use another one too, the one where there's like the Earth and the Moon and the Sun. But let's start with this one. So in the yellowish, because the flooding is not that intense, let's say from 0.5 to 1 in this type of reference for the water level. This is a type of reference, the mean lower water. But just ignore those names and just think that this is like a point of reference for how much water it's, let's say, on top of the city. So if it's closer to 0.5, it's going to be very minimal touching the streets, and it's probably only going to last like a few minutes, 10 something minutes. And then when it becomes closer to 1, of course, the affectation is higher, and also the time that it takes for that water to retreat and leave those streets increases. And then if you move to the next level, the red dots, then also the affectations and the time that the water stays, it increases. And it only happens, for example, for our event, let's say occurs in a Friday, like this flooding event, sampling event with the volunteers occurs in a Friday. That is the highest astronomical tide that we can predict. But before those days, there is also flooding happening. And after those days, because that's going to be like the highest point, like the push astronomically is going to be like the highest. But at this point, there was also a big push, and the day before, there's still a big push, not less, not as much, but definitely good, and so far. So before and after, there is still affectation depending on the combination of those factors.
Thank you for answering that question. While we wait for other questions, oh, actually, we just got one here. I'll put my question to the side. So this next question asks, what follow-up questions are you most interested in for this work based on what you've learned so far?
Yeah, definitely. Now we just don't want to focus. I'm gonna show you a graphic. That's what I'm doing here. We don't want to focus just in the one flooding event, or in this case 5 flooding events, but we want to try to project that, let's say, throughout the year, and probably throughout other regions in the Chesapeake Bay, or probably other regions of the world. So what we're doing is trying to get the data that we have collected for the past years, and try to use some land use and land coverage information, and trying to find relationships between nutrients and land use. Let's say, like the example that I was giving at the beginning. If it's a grassy area, does it has more dissolved inorganic nitrogen or less? Like if it's a pavement area, does it have more or less? If it's like a residential area, like those type of components, we're trying to associate and try to build the association. And if associations are found, we're going to try to build a model with that and project this temporally and hopefully also spatially.
Excellent, thank you. Well, here's a compliment and a question. This person says, great talk. This is really interesting research. With sea level rise being something that is going to be hard to control / prevent or / decrease in the short term, what are things that can be done to minimize the nutrient inputs from flood events in the shorter term?
Yeah, that's not an easy one. I think first I will point out like doing more research, because even we have done 5 years, we have not been able to find other research directly associated with this. So my first answer, there is not enough information to know like, oh, one logical answer to me would be like, oh, stop fertilizing your lawns like a lot. Some of the sampling that we do when we go to the residential areas, you can see fluorescent lawn areas, and when the flooding is touching, you can see a lot of the stuff coming out of it, getting in contact with the floodwater. So it seems like a very logical, very easy connection, but then when we actually analyze the data, it's not as direct. The relationship are not coming as clean as that. So first I will say more research on this to actually figure out where the affectations are coming from. If these numbers, even the lowest scenario numbers, are very dramatic in terms of nutrient input. So if those are true, we need to figure out where are they coming from to try to do something. So yeah, I will say first more research, and then once we found those correlations, those relationships, we can act on them.
Okay, thank you. This next question says, great job. Are there additional water quality parameters that you hope will be included in the future for this water quality monitoring data set?
Yeah, one that I didn't have much time decide to cut for time reasons was the ENTEROCOCCUS results. Again, that's not at all the focus of my research or my expertise, but for collaborations, because the organization providing the funding is the Hampton Roads Sanitation District. They were interested in collecting samples related to fecal indicators. In this case, we're using ENTEROCOCCUS abundance. So the EPA established this limit for, I think it's like swimming or recreational water. So this is the limit, 104, most probable number, which is the unit used in this case, for 100 milliliters. So this limit was only found, as you can see, in the different years. Very few of the samples were under this limit. The rest of the samples were higher, and in some cases even like orders of magnitude. This darker color are 10,000s or higher, 10,000 most probable numbers per 100 milliliter. We found even like 24,000, which is the detection limit for the method. We don't really know for those samples, like even how concentrated the fecal indicators can be, because yeah, it's just reached the maximum possible for the method, and we didn't have other samples to repeat. But this, I think, would be something that it would be really important to pay attention. We have a lot of people getting in contact with that water to get to their houses. Like we have found, again, all type of pictures of kids playing in it. So we should pay a little bit more attention also in the human health aspect of it.
Excellent. We'll wait another second to see if there are more questions from the audience, but I just was really curious about the citizen science app that you mentioned called Sea Level Rise that the volunteers used. Who built that?
I'm pretty sure that it was through Wetland Watch, a nonprofit located in Norfolk. But I'm not sure like which institution, who they contacted to build that. But they are the ones that developed it, and they had this project called Catch the King, which is also related to flooding. Instead of the water quality piece, is the correcting the models, the inundation models. So people go with that app, walk literally around the edge of the flooding and drop pins every time, every, I don't know, 2-3 steps or something like that. And then that creates like a layer of like, this is actually what the flooding was. And then people working on those models can use that to correct. One of the papers that I was showing, it's exactly that, using some of that data to correct the flooding models, to using that data, using the volunteers' data.
That's a very interesting topic and something that a lot of people are asking about these days at the library. All right, so it looks like we may have gotten to the end of our questions. So I really want to thank you so much, Alfonso, for your presentation. It was very interesting. Oh, wait 1 second. We did get a question. The last question that just came in. Thank you very much for the great information and presentation. Have you done any action to spread this message to the public, especially younger generations or school students?
Yeah, I should have added to the supplementary slides that I have here some of those images, but one of the pictures, for example, that I was trying to use for the for the flyer for the invitation to the talk, it was a Lego representation that someone did of our event. So that is just one example. We currently still have, I'm pretty sure, if not, it just finished a presentation in a museum, for example, associated to other sea level rise topics, but we were also there. Like this project, Measure the Muck, was part of that. We have had participation in a podcast. We have had we have intentionally tried to put ourselves there because we think it's a very relevant topic with not enough attention. But then also the volunteer piece has called us to come and talk to. It was like some garden organization in Norfolk. They wanted us to come and talk to them. The turf grass also industry at some point asked us to come and give a talk in one of their conferences. So intentionally we have tried to do some, but then we have also got questions from some of our volunteers or indirect, or people who has known for us through unknown ways.
It's always amazing to find out how people learned about you, and hopefully people will learn about you through this presentation, which I hope gets spread wide and far. But thank you again for joining us today on the Knauss Lunch and Learn seminars. I hope you have a fabulous year at the NOAA Office of Education. And Teresa, I know you're out there. Thank you as well for joining us today and helping with the introductions all the way from EPA. Good luck to you during your Knauss Fellowship as well. And thank you, of course, to the Lunch and Learn committee that helps organize this. We hope at NOAA Central Library that you will join us again next month for another Knauss Lunch and Learn webinar. We host them every third Thursday of the month at 12 p.m. Eastern Standard Time. And you get to learn more about what this fabulous group of fellows are working on. So Alfonso is the first in his cohort, but he won't be the last. Excelente charla, Alfonso. Felicidades, aprendices. De nada. Have a wonderful rest of the day, everybody, and thank you again. Take care.